So let's take a look at 5.10. So in 5.10, let me pull up the question. So we are working with plants now, and fix my pen, sorry. Okay, so colored allele requires a big R allele. Colored allele, and I don't really, I'm not really sure exactly what allele is. Something inside a kernel, a corn kernel. So little r is recessive, of course, and it's colorless, so it leads to colorless allele. I don't know which is better. I probably would prefer the colored allele myself. But okay, let's look at the other gene. So big Y causes the plant to be green, and little y causes the plant to be yellow, and big Y is dominant to little y. So the question is, we have a plant of unknown phenotype and genotype, and we're going to cross that, or in the someone's imagination, this plant was crossed to a plant that has 2 little r's and 2 little y's. So colorless and yellow. Now the progeny of this cross right here, so we have the phenotypes in numbers of those progeny. There are 200 progeny, 88 of them, let's see, so 88 of the progeny from that cross were colored in green, 12 were colored in yellow, 8 are colorless and green, 92 are colorless and yellow. So we want to figure out the genotype and phenotype of this plant here, and we want to figure out the arrangement of alleles, you know, if it's heterozygous, on the chromosome, on the chromosomes, if those alleles are linked. You know, we need to figure out the arrangement of those alleles. So let's take a look at try to figure out the genotype first. Just what are the alleles in this unknown plant. So a colored in green offspring here would need to have at least one big R and one big Y, because big R is dominant to little r, big Y is dominant to little y. So colored in yellow, we'll need to have a big R and a little y, and no, okay, I should erase that right there, sorry. We need to have at least one big R and 2 little y's. So colorless in green, well, those plants need to have 2 little r's and at least 1 big Y. And colorless in yellow is 2 little r's and 2 little y's. So we know, we put this up here, so the plant we crossed to, the known plant, is homozygous recessive for both of these genes. So each one of these progeny have to have a little r and a little y. So let's assume all of these alleles here came from the known parent, the parent with the known genotype and phenotype. So that leaves all the others to have come from the plant with the unknown genotype. And we have a big R and a big Y here. And then in these cases over here, we have a little y that must have come from the unknown, and a little r over here that must have come from the unknown. So we know the genotype of the unknown plant has to be big R, big Y, little y. So I'm going to make some more room down here. And we figured out that the genotype of the unknown plant is, I'm going to change it back to yellow. I think you can see that better, right? So the genotype of the unknown plant is big R, big Y, little y, and the phenotype is going to be colored and green, the dominant traits. So this plant can make at least 4 types of, well, it can make 4 types of genetically distinct gametes. So big R and big Y can be together in one gamete. Big R and little y can be together in one gamete. Little r and big Y, and little r and little y. So, and we're going to, during the cross here, all of these are going to combine with that little r and little y. Now, if these genes R and Y are on different chromosomes, then they would be assorting independently, and we'd see roughly 25% of each of these gametes being produced in this plant. And if that were true, then we would see approximately 25% of these genotypes in the progeny. So big R, big Y, little r, little y, big R, little y, little r, little y, and so on and so forth. And so all of these, all the 4 phenotypes we saw in the progeny, I can scroll up here, all these guys up here, they should be seen in equal numbers. Now we don't see that, right? We see 88 colored green and 92 colorless yellow. So, and we only see 12 of colored yellow and 8 of colorless green. That means we are dealing with a case of linkage. So the opposite of independent assortment is gene linkage. So in this case, these genes are linked and their alleles are not assorting independently. So I want to erase something. I'm not really sure which thing to erase to make this easiest to show the solution, the easiest way to find the solution here. Okay, so maybe the best thing to do is for me to scroll down here and reiterate that we know the genotype of the unknown plant is big R, big Y, little y, and we know the genes are linked. So there are 2 possible arrangements of alleles in this plant. So we can have the big R and the big Y on one chromosome, and the little r and the little y on the other chromosome. So that's one arrangement. And the other arrangement is big R with little y and little r with big Y. So there are no other possibilities here. And we need to determine which arrangement is the correct one just by looking at the numbers of the progeny of the offspring. So I think maybe the best thing for me to do is Can I copy and paste this? Let's see if I can copy and paste this. I'm going to select, try copying that. Let's see. Can I shrink it? Cannot shrink. Oh, I can shrink it. Nice. Okay, let's see here. Let's see. Hope you can still see this on your screens. Okay. So you can, let's take a look here at this 88 colored in green. So 88 there colored in green and 92 colorless in yellow. So the most predominant phenotypes or genotypes in the progeny are, no pun intended, big R, little r, big Y, little y. Remember, these are coming from the test cross parent, the homozygous recessive for both R and Y parent. And this genotype down here, little r, little r, little y, little y. So these two groups of offspring right here, the highest representative phenotypes in the offspring, inherit either a big R and a big Y together from the unknown parent, which is known now, or a little r and a little y from that parent. So these two, the big R is most often with the big Y, and the little r, in the gamete I should say, from the unknown parent, and the little r is most often with the little y in the gamete from the unknown parent. And we also know that non-crossover gametes always outnumber crossover gametes. So these gametes right here have to be the non-crossover gametes from the unknown parent. And the only way for those to be the non-crossover gametes are for this to be the correct arrangement of alleles in the unknown parent.